Have you ever tried to assemble a really complex piece of furniture only to realize the box is completely missing the instruction manual?
Oh, yeah. The absolute worst.
Right. You have, all the parts, all the screws, the wooden panels, but you're left just kind of guessing at how they actually fit together.
And usually you end up with something that vaguely resembles a bookshelf, but you definitely wouldn't trust it to hold any heavy books.
No, absolutely not. The structure is there, but the integrity is just completely compromised.
Exactly.
So keeping that in mind, imagine that unstable bookshelf is actually a human kidney grown in a laboratory. And instead of holding books, it's supposed to filter your blood.
Which is terrifying if it's falling apart.
Yeah, exactly.
Yeah.
So welcome to this deep dive. Today, we're looking at this really incredible 2026 paper published in the journal Science. And our mission here is to explore this quest to build miniature, lab-grown human kidneys.
Right, which biologists call organoids.
Yes, organoids. But more importantly, we're going to unpack how scientists finally figured out how to give these cellular building blocks their missing instruction manual. Because without it, they were just building this chaotic, tangled plumbing.
It really is a phenomenal leap in synthetic biology. I mean, we're moving from simply observing biological chaos in a petri dish to actively engineering spatial order.
Which is huge.
It is. We're essentially learning how to draft the blueprints that the cells have, been missing this whole time.
So let's start by unpacking the baseline here for you listening. What exactly is a kidney organoid? You can think of human stem cell-derived kidney organoids almost like this highly talented jazz band playing entirely without sheet music.
That's a really good way to look at it.
Right, because these stem cells are incredibly capable. They self-organize into real kidney tissues, which is amazing in its own right. But the architecture they build is totally unpredictable.
Yeah, it tends to be radially symmetric.
And for those of us who aren't biologists, that basically means it forms a uniform, round sphere of tissue, right?
It's exactly, just a ball. And if we connect this to the bigger picture, that radial symmetry is a massive biological roadblock.
Why is that?
Well, in a real human kidney, the primary filtering units, which are called nephrons, they can't just form a random ball of cells. They have to form these long, highly segmented tubes.
Right.
And those tubes must be perfectly oriented in a very specific direction.
Because of the fluid dynamics, right? I mean, they're filtering waste, and that fluid has to go somewhere.
Precisely. The fluid they filter has to drain into a common central pipe known as the collecting duct.
Okay.
So if the organoid nephrons lack spatial organization, if their plumbing points inward or outward or just tangentially in random directions, they cannot form a coherent draining system.
But pipes just don't connect to the main sewer line.
Exactly. And that severely limits our ability to use these organoids for modeling complex kidney diseases or, looking toward the ultimate goal, transplanting them into human patients to replace failing organs.
But wait, I want to push back on that a second. I thought we already figured out how to trigger these stem cells to grow. Don't we just give them a chemical nudge to start the process? Yeah. Because researchers have been growing these for a few years now.
We do, yeah. We know how to start the process, but starting it and guiding it are two very different things.
Oh, I see.
Currently, biologists use a chemical pulse. It's called the Takasato protocol. On day 7 of the stem cell's development, they're hit with a drug called a GSK3 inhibitor.
And that's specifically a molecule called CHIR 99021, right?
Yes, exactly, CHIR.
And what exactly does that chemical pulse do to the cells? What's the actual mechanism there?
It acts entirely like a starter pistol at a race. It mimics the early WNT signaling proteins needed to induce the birth of the nephron.
So it kicks things off.
Right. And if we track the timeline of this growth, that CHIR chemical pulse triggers a cascade. By day 10, those undifferentiated cells condense into tight, organized clusters called protubular aggregates, or PTAs.
Okay, PTAs at day 10.
Yep. Then, by day 12, those clusters hollow out to form these fluid-filled structures called renal vesicle-like nephrons, or RVs.
So they are genuinely transforming into kidney tissue at that point. They have all the right cellular machinery.
They absolutely do. They express the correct structural proteins and genetic markers. You'll see things like CDH1, JAG1, and WT1 expressing perfectly. They have successfully become kidney cells.
That sounds like a success, though.
Well, the problem is that this chemical pulse is just a transient global signal. You're basically bathing the entire petri dish in this chemical.
Oh, so everything gets hit at once.
Exactly. It screams, grow a nephron to every single cell simultaneously, but it provides provides absolutely zero spatial direction.
So it tells them what to do, but not where to go.
That is the perfect way to phrase it. There is no compass. The cells wake up. They know they're supposed to be a kidney, but they have no idea which way is up, down, left, or right relative to each other.
Man, that's fascinating. So let's just do a quick 5-minute recap for you listening to make sure we're all on the same page before we get into the really crazy stuff.
Good idea.
So bullet point 1, organoids are miniature stem cell-derived organs that self-organize, which is awesome. Bullet point 2, current kidney organoids do form filtering nephrons, but they are uncoordinated and radially symmetric, just confused little spheres.
Right.
And bullet point 3, chemical triggers like that CHIR starter pistol start the growth process, but they don't provide a map. So this brings up a really big question for you to think about. If you were a stem cell that has just been told to build a kidney, but you don't know which way to orient yourself, where in the world do you look for a compass?
And to answer that, the researchers realized they had to step away from the petri dish entirely.
They had to look at the real thing.
Exactly. They had to figure out what these lab-grown organoids were missing by going back to the original source. They mapped out actual, naturally developing human fetal kidneys.
And this was specifically looking at that critical window between weeks 13 and 17 of development, right?
Yes, that's the key window.
And to do this, to actually map this out, they used a relatively new technology called spatial transcriptomics. Let's break this down because the way this works is mind-blowing.
It is incredibly powerful. They use specific platforms known as Xenium and Seqofesh.
Seqofesh.
Yeah, it stands for sequential fluorescence in situ hybridization.
Okay, so instead of the old way of doing things, where you basically, grind up a piece of tissue in a blender to see what genes are present.
Right, the old bulk sequencing method.
Yeah, which is kind of like looking at a fruit smoothie and trying to guess the original shape of the fruit.
Yeah.
Instead of that, this spatial transcriptomics technology is more like Google Earth for cells.
That is a great analogy. It allows researchers to take a pristine, intact slice of tissue and attach fluorescent tags directly to the mRNA molecules inside the cells.
So under a microscope, the tissue literally lights up.
It does. You can see exactly which genes are turned on, and you can see their exact physical GPS coordinates within the architecture of the kidney.
Which is insane. And when they looked at this cellular Google Earth, they zoomed in on that main drainage pipe you mentioned earlier, the collecting duct.
And what's fascinating here is what they found residing in that duct. They discovered a crucial developmental organizer. There was a very sharp, highly localized signaling boundary right within the tissue of the collecting duct itself.
Okay, let's create a visual mnemonic for this. Imagine the collecting duct is like a plant. The pointy tip of the leaf expresses a protein called WNT-11.
Right, WNT-11 at the tip.
And the sturdy stalk, or the base of the plant, expresses WNT-9b. So just remember, 11 is the pointy tip, 9b is the base, the stalk.
I like that. And it's that WNT9B stalk area that acts as the critical beacon.
Let's use another visual here for you listening. Imagine you're standing near a warm campfire on a really cold night. The side of your body that is facing the fire gets warm, right? But the side of your body facing away into the dark stays cold. Your body suddenly has a physical axis based entirely on your proximity to that heat source.
That campfire dynamic is exactly what is happening in the collecting duct, but, with chemical proteins instead of heat. The researchers found that the duct secretes that WNT9B signaling protein, and the nascent, newly forming nephrons in the fetal kidney always cluster right next to this WNT9B source.
So the WNT9B is the campfire.
Yes. And just like your body near the fire, this creates what researchers termed an adjacent to distant axis, or AD axis. The single-cell and single-nucleus RNA sequencing proved that the cells in the collecting duct are literally broadcasting a chemical signal to the nephron cells right next door.
Okay, wait, I want to challenge that assumption for a second. How do we know it is genuinely a chemical broadcast that shapes the nephron?
What do you mean?
Well, could it just be that the nephrons are physically squished up against the collecting duct, and the mechanical pressure, the actual physical crowding of the cells, is what causes them to organize?
That is a really critical distinction to make, and it's exactly why they look so closely at the genetic switches inside the cells. If it were just mechanical squishing, you wouldn't see a massive, targeted rewiring of the cell's genetic code. But the sequencing showed us the genetic switches flipping in real time.
Okay, so what kind of switches are we talking about? Give me the granular details here.
All right, so when a cell situated directly adjacent to the collecting duct is bathed in that high WNT9B protein, it turns on very specific markers. We're talking FGF8, LHX1, JG1, and EMX2.
The whole alphabet soup.
Exactly. But the most important one that flips is WNT4. WNT4 is a classic canonical WNT target gene.
And what does turning on WNT4 actually do?
When WNT4 turns on, it locks in that cell's identity. It chemically programs that cell to become the connecting piece of the pube. Meanwhile, the cells distant from the duct, the ones on the cold side of the campfire, turn on completely different genes like MEOX1 and PCDH15.
Wow, okay, so this is the exact moment biological symmetry breaks.
Yes. The nephron is no longer a uniform sphere of identical cells. One side is now chemically and fundamentally different from the other.
And as it elongates into these comma and S-shaped bodies, that AD axis guides the classic proximal distal axis.
Right, with distal markers like GATA3, POU3F3, and TFA2A enriching right near the collecting duct.
So the WNT9B signal isn't just a trigger to grow. It is the physical beacon that tells the nephron which way to face.
Precisely.
All right, time for our next quick recap. Bullet point 1, spatial transcriptomics maps exact gene locations in real fetal kidneys, like a cellular Google Earth.
Yep.
Bullet point 2, a WNT11 and WNT9B boundary in the collecting duct acts as a local signaling beacon or campfire. And bullet point 3, nephron cells close to the beacon turn on different genes than cells far away, forming that adjacent to distant axis and breaking the symmetry.
Spot on.
So here's the question for you to ponder now. Think about that campfire. What happens to our lab-grown organoids if we remove the fire entirely?
And that is the exact dilemma of the organoid. When the researchers returned to the lab and checked the RNA of their lab-grown tissues, they found that the campfire was completely missing.
Just gone.
Totally. The WNT9B gene wasn't just low, it was totally absent. Other WNTs, like WNT3A, were missing too. The organoids were trying to build a draining system without the central beacon.
And consequently, that crucial genetic switch you mentioned, WNT4, that must have been drastically reduced in the nephron cells.
Exactly. It was practically turned off.
But did the organoid cells actually lose the ability to hear the signal? Like, did they become deaf to it, or is the signal just not there?
This raises a really important biological question of competence versus source. Are the cells incompetent, or is the environment failing them?
Right.
The researchers looked at the cell surfaces and found that despite lacking the WNT signaling proteins themselves, the organoid nephrons still possessed all the necessary WNT receptors.
Okay, what kind of receptors?
Specifically, they had proteins called frizzled receptors, FCD4, FCD5, FCD27, and LRP56, sitting right on their cell membranes, waiting to catch a signal.
Okay, I love this. So they have the radio antennas installed and wired perfectly on the roof, but the local radio station is totally offline and not broadcasting.
Exactly. The cells are fully competent, they just lack the source. So the researchers had this massive aha moment and decided to play architect. They set out to build a synthetic radio station from scratch.
This is the crux of the synthetic biology intervention in the paper.
It is. They engineered human embryonic kidney cells, often called HEK cells, to act as synthetic organizers.
The engineering behind this is wildly elegant. Let's break down mechanically how they built this synthetic radio station, because they didn't just dump a chemical into the dish this time like the Chi-HR pulse. They built a localized beacon.
Right, they had to keep it local.
So first, they gave these engineered HEK cells a gene called cadherin 3, or CDH3. Cadherins are basically proteins that stick out of the cell membrane and act like cellular Velcro.
A great way to describe it.
By giving all these synthetic cells the same Velcro, it forced them to clump tightly together into a tiny, dense sphere, rather than just floating away into the fluid.
Which is vital for keeping the signal localized in one spot, rather than washing over the whole dish.
Right. Second, they gave them a fluorescent tag called M-Sherry, which means these clumped cells literally glowed bright red under a microscope.
Which allowed the researchers to track their exact physical location in real time.
And finally, the actual payload. They engineered these red Velcro clump cells to continuously manufacture and secrete WNT3A.
Then came the moment of truth.
Yeah, set the timeline up for us.
Okay, so they took these red, glowing, WNT-secreting spheres, the synthetic organizers, and dropped them right next to the non-polarized day 12 organoids.
Remember, these are the organoids that had just formed those confused, radially symmetric spheres of cells we talked about.
Exactly. They physically placed the synthetic campfire right next to the cold cells.
So what happened by day 14?
The radio station was turned on, and the frizzled antennas picked it up immediately. The WNT targets inside the organoid nephrons genes like LEF1, EMX2, FGF8, DKK1, and DLL1. They absolutely spike.
But far more importantly than just gene expression, the physical architecture changed, right?
Yes. The radial symmetry finally broke. The organoid cells actually realized which way was up. They observed that the early distal to medial marker, JG1, increased specifically on the side of the cell cluster closest to the synthetic organizer.
Wow.
Meanwhile, the proximal marker, WT1, was reduced near the organizer. The nephron completely polarized itself, physically orienting its growth relative to the new synthetic beacon.
Now, you know in science, correlation is not causation. Just because they dropped a glowing red ball next to the organoid and it polarized, how did they prove it was specifically the WNT broadcast causing the alignment?
Right. Couldn't it be something else the HK cells were doing?
Ex-
The researchers anticipated that exact skepticism. To prove the mechanism, they introduced chemical inhibitors into the petri dish, specifically drugs called IWR1 and IWP2. Plus, they used a yellow fluorescent proxy, HNF4A-YFP, to track the proximal precursors.
Okay, but what do those inhibitors actually do?
They basically block the WNT pathway from functioning. They essentially put earplugs in the organoids.
And let me guess, when they put the earplugs in, the cells went back to being confused spheres.
Completely. When they blocked the WNT pathway, the polarization stopped dead in its tracks. This proved definitively that the chemical broadcast from the synthetic organizer was the direct mechanical cause of the symmetry breaking.
This is such a massive leap. All right, time for recap number 3. Bullet point 1. Organoids had the antennas, the frizzled receptors, but they lack the WNT broadcast. Bullet point 2. Scientists engineered synthetic organizers, or SOs, to act as a localized WNT beacon using cellular Velcro and a red glowing tag. Bullet point 3. When placed next to organoids, these SOs successfully broke cell symmetry, polarizing the nephrons and proving the concept.
It's beautiful engineering.
It really is. But this brings up a very important question for you listening at home. We've given ourselves a compass, and they are following it. But if one WNT speaker is good at giving directions, is a stadium-sized rock concert speaker better? Like, how loud should this biological broadcast actually be to build the perfect tube?
And that brings us to a foundational concept in developmental biology called the morphogenetic field. In biology, more is almost never better.
That's all about balance.
Exactly. It's all about gradients. The researchers needed to figure out how to control the volume of the signal to find the WNT signaling Goldilocks zone.
And to control the volume, they didn't just, add more cells, right? They engineered a volume dial directly into the DNA using a drug called doxycycline.
Right.
How does that mechanism actually work?
It is a brilliant piece of genetic wiring. They created inducible organizers, I13SO and IWNT9BSO.
Okay.
They took the WNT gene inside their synthetic organizers and attached it to a specific genetic promoter. You can think of a promoter as a toll booth on the DNA.
A toll booth?
Yeah. This specific toll booth only opens, allowing the gene to produce WNT protein when the drug doxycycline is present in the culture. Oh, and for the WNT9B organizers, they also needed to add a protein called RSPO1 to potentiate the signal.
So by adding different, highly controlled concentrations of doxycycline into the petri dish, they could literally dial the WNT output up or down.
Exactly. It produced a beautifully predictable sigmoidal dose-response curve.
Amazing.
Once they had this volume knob, they let the organoids grow out to day 18 to see the final architectural results under different volumes. And this is where the reality of the morphogenetic field becomes visible.
So let's look at the 3 main outcomes they tested at day 18. Scenario 1, high WNT. They used a heavy dose of 50 nanomolar doxycycline. This was the rock concert speaker. I'm guessing a signal that loud completely overwhelmed the system.
It did. Biologically, the nephrons became what we call overly distalized. Remember that the nephron is a long tube with different identities at each end.
Right.
When the WNT signal was blasted at high volume, the cells directly under the organizer formed massive connecting distal structures. But the proximal structures, the delicate filtering units called renal corpuscles at the far end of the pipe, they were almost completely suppressed within a 90 to 100 micrometer radius.
Because the campfire was so hot, the cells thought they were all standing right next to the WNT source, so none of them developed the distant identity.
Precisely. The signal was so loud it drowned out the spatial gradient necessary for the other cell identities to form.
Okay, so scenario 2, minimal WNT. Essentially turning the dial down to 0 nanomolar dox.
With the volume off, the growth reverted to being largely stochastic.
Just random.
Yeah, random orientation, just like the baseline unmodified organoids. The cells mostly retained their proximal structures, but they lacked any coherent organization or elongated tube structure.
Which brings us to the third scenario, the Goldilocks zone. They used an intermediate volume of 5-nanometer doxycycline. What happened to the architecture here?
This is where the magic happened. At this intermediate volume, the morphogenetic field was perfect. The nephrons generated multiple continuous segments. They formed the distal segments, the proximal tubules, and the renal corpuscles.
Wow.
But the crucial difference was they were no longer random spheres. They elongated properly.
So they actually looked like real kidney plumbing.
They remained beautifully elongated and coherently oriented. Their proximal distal axes physically aligned toward the WNT source. They all pointed their distal ends, the exact ends of their tubes that need to connect to a collecting duct, directly at the synthetic organizer.
That is staggering. All right, our final quick recap, bullet point 1. Scientists made the synthetic organizers tunable using that doxycycline toll booth. Bullet point 2. Too much WNT suppresses proximal cell identities. Too little leaves them completely unorganized. And bullet point 3. An intermediate WNT gradient perfectly elongates, patterns, and aligns the nephron tubes, which proves you don't need the whole tissue, just the geometry of the signal.
Exactly.
So synthesizing all of this for you listening, we've essentially hacked the biological instruction manual using just one localized signal. This deep dive shows us that the magic of complex fetal development isn't always about having exactly the right mixture of thousands of different cells all perfectly touching each other.
Right.
Sometimes it is just about pure geometry. By simply placing a synthetic WNT broadcaster in the right spot at the right volume, the organoid's own stem cells knew exactly how to organize themselves into working architecture.
And that emphasizes a massive, broader implication here. This pipeline mapping a tissue's native spatial signaling and then synthetically reconstructing just that specific geometry is a modular strategy.
It's plug and play.
Essentially, yeah. It bypasses the need to rebuild the entire native environment like building a whole collecting duct from scratch. We just provide the WNT beacon, and the stem cells do the rest of the heavy lifting.
It's like finding a massive shortcut in biology. You don't need to read the entire encyclopedia if you just know exactly where the most critical screws need to go.
Exactly.
Which leaves you with a final provocative thought to mull over today. If we can hack the architectural blueprint of a human kidney just by discovering and replicating its local WNT boundary, what other organs are waiting for us to discover their secret geometric beacons?
It's a huge question.
Could a single engineered beacon, just a tiny glowing cluster of cellular Velcro, be the key to eventually growing a perfect transplantable heart or a liver in a lab? Think about that the next time you hear about a breakthrough in stem cells. It might not just be about the cells themselves, but the invisible spatial beacons guiding them in the dark.
A really exciting frontier.
It absolutely is. Until our next deep dive, keep questioning the blueprint.